Hello guys, in last video we had discussed that discovery of cell and shape and size of the cell and structural organization of a cell and cellular composition in different organisms and about the most important topic, that is prokaryotic cells and eukaryotic cells. So in today's class we are going to start with cell organelles. So what are cell organelles? So for a cell, we need a lot of chemical activities to support their complicated structure and functions. So we have learned the structure of cell, right? So their structure is really very complicated. Our body is made up of organs. So what are organs made up of? They are made up of cells. So inside cells we have organelles. So we are going to study in this class about cell organelles. So structures which perform specialized functions within themselves are called cell organelles. So we have few cell organelles. Those are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. So we are going to study about all the cell organelles. So you can see the pictures of the cell organelles, right? So you can see the endoplasmic reticulum, Golgi apparatus, lysosomes, and mitochondria, and plastids and vacuoles. So coming to the next part. Yeah, now we are going to study about the first organelle, that is endoplasmic reticulum. So this endoplasmic reticulum, it is a network of interconnecting membranes distributed throughout the cytoplasm. So if this is a cell, and in center we are having a nucleus and the other part is cytoplasm. So almost the cytoplasm is covered with endoplasmic reticulum. So the endoplasmic reticulum membrane is similar in structure to the plasma membrane. It is almost similar to the plasma membrane. So this endoplasmic reticulum is of two types. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. Rough endoplasmic reticulum is nothing but RER and smooth endoplasmic reticulum is SER. So now we are going to study about rough endoplasmic reticulum. So this rough endoplasmic reticulum, why the endoplasmic reticulum is rough here? Because it contains ribosomal particles on its surface, due to which its surface is rough. So you can see the picture here. On its surface there are small dots. You can see those are ribosomes. So that's why its surface is rough. So the ribosomes are the site of protein synthesis. So these ribosomes, you can see it here. So these ribosomes help in the synthesization of proteins. So RER is mainly formed by crystallinae. So this rough endoplasmic reticulum is mainly formed by crystallinae. So coming to smooth endoplasmic reticulum. So what is the smooth endoplasmic reticulum? As the rough endoplasmic reticulum looks rough because due to the presence of ribosomes on its surface. So smooth endoplasmic reticulum, its surface is smooth due to the absence of ribosomes. So the endoplasmic reticulum, it is smooth endoplasmic reticulum because it does not have ribosomes on its surface. It helps in the manufacture of fat molecules or lipids. So it helps in the manufacturing or synthesization of lipids or fat molecules. It is formed by vesicles and tubules. So vesicles and tubules, these are formed by smooth endoplasmic reticulum is formed by vesicles and tubules. So the function of endoplasmic reticulum. Now we are going to study about the function of endoplasmic reticulum. So protein synthesis is done by rough endoplasmic reticulum. We had studied that the synthesis of protein is done by rough endoplasmic reticulum, and fats and lipids are synthesized by smooth endoplasmic reticulum. So protein and lipids synthesized on ER are used for making cell membrane. So now we can see the fact that the membrane, the cell membrane, it is made up of proteins and lipids. So always the cell membrane is made up of proteins and lipids. So the SCR is in liver, takes part in deoxifying many poisons and drugs. So the main function of SCR is it is present in liver cells, which help in the deoxifying many poisons as well as drugs. So now we are going to study about Golgi complex. So this Golgi complex is also known as Golgi apparatus. So it was first described by Canelo Golgi. So at the name only you can see that Golgi is given to the name by Canelo Golgi. So it consists of system membrane bound vesicles. So system membrane bound vesicles is nothing but flattened sacs. So you can see the Golgi apparatus. They are flat here, right? So they are arranged parallel to each other. So they are arranged parallel to each other. Parallel means beside to beside. So you can see the picture here. They are arranged parallel to each other. So in a cell, Golgi apparatus looks like this. Now, the functions of Golgi complex. So what is this Golgi complex function? So storage, modification and packing of products. So the main function of this is storage, modification and packing of products. It breaks down complex sugar into simple sugar. So the complex sugar is broken down into simple sugar. So it helps in the formation of lysosomes. So lysosomes is also a cell organelle which we are going to study in the next slides. So it is involved in the synthesis of cell wall and plasma membrane. So it is involved in the formation of cell wall and plasma membrane. So coming to lysosomes, so these are produced by Golgi apparatus. We had learned in the functions of Golgi apparatus or Golgi complex. So this is a cell organelle which is like it helps to keep the cell very clean. So it is a waste disposal system of the cell. So it is a waste disposal system of the cell. So lysosomes are membrane-bound sacs that are filled with digestive enzymes. So it is a bag or a sac which is filled with digestive enzymes. So first of all, what is this enzymes? So enzymes are made by rough endoplasmic reticulum. So these are the digestive enzymes are made from rough endoplasmic reticulum. Lysosomes are also called the suicide bags of a cell. So do you know why these lysosomes are called the suicide bags of the cell? Because suppose a foreign material enters the cell, that time this lysosome bursts out and inside the lysosomes the digestive enzymes start roaming in the cytoplasm and makes the foreign material go away. So when there is a disturbance in the cell, so if the cell damages, the digestive enzymes, it could digest the whole cell itself. So it is so powerful. Okay, now we are going to discuss about functions of lysosomes. So they help to keep the cell by digesting any foreign material that enters the cell as well as worn out cell organelles. So as I had told, it helps to keep the cell very clean by digesting any foreign material. Okay, so it breaks down complex substance into simpler substance. It breaks down complex substance into simpler substance. So one of my favorite cell organelle is mitochondria. So this mitochondria, it was first observed by Kolliker in 1880. In 1880, it was observed by Kolliker. So it is a double membrane bounded cell organelle. So it is a double membrane bounded cell organelle. So the outer membrane is very porous, as we had studied that it is a double membrane cell organelles. The outer membrane is porous and the inner membrane is deeply folded with finger-like projections called cristae. So you can see in the picture here. So these are the finger-like projections called cristae. So you can see here cristae. So it creates a large surface area for ATP, that is endozyme triphosphate. That is endozyme triphosphate. generating chemical reactions. So this endozyme triphosphate is a chemical reaction which is produced by mitochondria. So the functions of mitochondria are it generates energy for various activities of cell. So it generates energy for activities of cell, whichever activity it may be, it generates energy. So it is like an energy booster for the cell. For us, the energy booster is food. So it is also known as the powerhouse of the cell. It is also known as the powerhouse of the cell. They release energy required by the cell in the form of ATP, endozyme triphosphate. As I have told you, this ATP is a chemical reaction which is released by mitochondria. So mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. So now we had learned about ribosomes in which organelle? We had learned about ribosomes in endoplasmic reticulum, that is under rough endoplasmic reticulum. So why does it makes the rough endoplasmic look rough? Because of the presence of ribosomes on its surface. So this mitochondria have their own ribosomes and DNA. Now the interesting organelle is plastids. So the internal organization of plastids contains numerous membrane layers embedded in a material called stroma. So this plastids is having many numerous membrane layers embedded in a material called stroma. So in a material called stroma, there are many membrane layers which are embedded. So plastids are similar to mitochondria in external structure. Seeing external structure, plastids are similar to mitochondria. They contain their own DNA and ribosomes. So they have their own DNA as well as ribosomes. So plastids are of three types: chloroplast, leucoplast and chromoplast. So coming to chloroplast, so this is a table of plastids. So chloroplast, chromoplast and leucoplast. So chloroplast looks like this and chromoplast look like this and leucoplast look like this. So we are going to study about chloroplast, chromoplast and leucoplast in the further slides. So functions of plastids. So coming to chloroplast, these are plastids containing chlorophyll. So you can analyze in the name itself. So plast means it is derived from the word plastids. Chlorophyll. So chlorophyll is nothing but a green pigment. So they are the plastids which contain green pigment called chlorophyll. So chloroplasts also known as kitchen of the cells. So they are also known as the kitchen of the cells. So they give a green color to the plant. They give green color to the plant. So the functions of chloroplasts are, these are important for photosynthesis in plants. So these are very important plastids which are important for photosynthesis in plants. So I would like to give a very important information. Plastids are only found in plant cells. In animal cells, plastids are not found. So coming to leucoplasts, these are the white or colorless plastids. These don't have colors only. They are the white plastids. They can change into the types of plastids. They can change their shape also. So the functions of leucoplasts are, leucoplasts store materials such as starch, oils and protein granules. So the main function is storage here. So they store oils and proteins. So the next function of chromoplasts is, these are colored plastids. So the uncolored plastid is leucoplast and the colored plastids is chromoplast. So except green, all the colors we can find in chromoplast. So where can green color be found? It can be found in chloroplast because they have a pigment called chlorophyll. So the function of chromoplasts are, they impart color to flowers and fruits. So they give color to flowers as well as fruits. They are rich in lipids. So they are rich in fats. So coming to the last cell organelle, that is vacuoles. So vacuoles, these are the storage sacs of solids or liquids contents. So these are the storage bags which contain solid or liquid. So in animal cells, vacuoles are small size. So in animal cells, they are very small in size. But in plants, the vacuoles are large sized. In plants, they are very large. Functions of vacuoles. So vacuoles are full of cell sap and provide turgidity and rigidity to the cell in plants. So they give turgidity and rigidity to the cell of a plant. So they give the shape of a plant. Many substances like amino acids, sugar, organic acids and proteins are stored in vacuoles. So these acids, proteins, lipids are stored in vacuoles. So in amoeba, consumed food items are stored in vacuoles only. So amoeba is a unicellular organism. So the food items are stored in vacuoles. In some unicellular organisms, vacuoles also play an important role in expelling excess water and some waste from the cell. So in unicellular organisms like amoeba, chlamydomonas and protozoa, these vacuoles perform a very important role in expelling the excess water or waste from the cell. So these are the vacuoles. You can see the vacuole here. Almost the plant cell is covered with vacuole only. You can see here, right? This is an animal cell. The vacuole is really very small. So this is also an animal cell. It is really very small. So thank you for watching our video. If you have not subscribed our video, if you are now not subscribed our channel, please do subscribe it. And thank you. Bye. Stay home. Stay safe.